As most people are aware, there have been enormous strides over the last 5 to 10 years in terms of new drugs for patients with T-cell lymphoma and the increasing awareness that these are very rare diseases that have remarkable heterogeneity with nearly 29 subtypes. What's happening is the pace of science is moving much faster than our ability to put patients on relevant clinical trials where we can ultimately answer questions regarding best therapeutic modality and understand a little bit about what kinds of patients are responding to these different treatments. And so with all of this rapid progress and the amazing science that we've heard about this weekend here at the T-cell Lymphoma Forum in La Jolla, there is a need to have more global harmonization in our ability to work together as one and agree on the important clinical trials that we need to conduct. And in that vein, we have assembled myself, Pierluigi Zinzani, representing the European sector, and Won-Suk Kim from Samsung Medical Center in Seoul, representing the Asian sector, and myself representing the North American sector, have gotten together using Columbia University as the coordinating site to try to orchestrate an international group zeroed in on doing the exact same clinical trial and trying to accrue that trial as expeditiously as possible so we can answer important questions and then realize what's the next important question to answer and then move on through a clinical trial-based approach to conduct those studies as efficiently and expeditiously as possible. So there's been a lot of work from my group and others looking at novel combinations. And so where I would say the last 10 years has been largely devoted to identifying new drugs active in T-cell lymphoma, I think the next 10 are going to be zeroed in on how to leverage the combinations, because there's really few to no cancers that we treat with single agents. We are always thinking about identifying complementary drug-drug interactions and study the combination. And so work from my group has established that many of these new drugs approved in T-cell lymphoma, like HDAC inhibitors, romidepsin, belinostat, pralatrexate, and a number of new biologics, actually are highly synergistic together. And so Salvia Jain from my group has looked at combinations of pralatrexate and romidepsin, and Enrica Marchi has looked at combinations with hypomethylating agents and HDAC inhibitors. And so we now have 2 studies that are opening imminently, where we're going to look at adding immune checkpoint inhibitors like pembrolizumab and or durvalumab from Celgene, and try to look at adding them to these different combinations and or the single agents to see if they represent an incremental improvement over the single or double agents that we've been studying. So one of those studies will be a 4-arm study looking at the addition of durvalumab to hypomethylating agents, HDAC inhibitors, and or their combination. And another, working with Merck, will look at pembrolizumab in combination with pralatrexate, decitabine, and the combination of pralatrexate and decitabine. Amongst the drugs that we know are active in the disease, it also looks like there are a number of other drugs like PI3 kinase inhibitors, of which there are now many, and proteasome inhibitors, and some interesting novel biologics. And given these mechanisms and some suggestion that these drugs as single agents have activity in T-cell lymphoma, we're now moving to explore and broaden the number of those doublet or triplet combinations we're going to be focused on by looking at combinations of PI3 inhibitors plus some of these drugs. There's strong scientific rationale I can't go into at the moment that is predicated largely on mechanism, but suffice it to say that the mechanism for many of these other novel combinations like PI3 kinase plus HDAC or XPO inhibitors like selinexor and HDAC inhibitors are extremely intriguing and I think going to represent the next wave with really the ultimate goal of trying to create 3 or 4 drug regimens with some added biologics to try to improve the activity and create something that might be akin to our CHOP-based chemotherapy, but won't be CHOP-based predicated treatment.